Vectors, plasmids, cosmids, phagemids, lambda, bacteriophage, M13, MAC, and DIAC. DNA encodes genetic information in all living cells. Genetic engineering is a branch of science that deals with the transfer of DNA between the hosts by in vitro enzymatic manipulation. Since most of the DNA fragments are incapable of self-replication in host cells, an additional segment of DNA capable of autonomous replication must be linked to the fragment to be cloned. This autonomously replicating fragment is known as a molecular cloning vector. They facilitate the stable integration and multiplication of exogenous DNA inside the host system. This technology is termed as molecular cloning. It involves series of sequential steps which include a restriction digestion of DNA fragments, both target and vector DNA, ligation of the target DNA with the vector, and their subsequent introduction into the host organism for multiplication. In general, vectors should possess some key features necessary for their function, capable of autonomous replication inside the host site, a compatible restriction site for insertion of DNA molecule or insert, presence of selectable marker for screening positively transformed cells, smaller in size and able to incorporate inserts of larger size. So types of cloning vectors, plasmids are there. Plasmids are naturally occurring extrachromosomal double-stranded molecular DNA circular DNA molecules that can replicate autonomously inside host bacterial cells. Plasmids range from about 1 to 250 kb in size. They are stably inherited extrachromosomally. They encode for only a very few proteins which are required for their own replication. These protein encoding genes are located very close to the origin of replication site. The rest of the needed proteins are provided by the host cell machinery itself. Thus, only a small portion of surrounding the site is required for its replication. Hence, the parts of a plasmid can be detected and foreign sequences can be inserted without compromising replication. The number of molecules of a plasmid present in a single bacterial cell is termed as copy number. It ranges from 1 to 50 per cell, but its number is specific for a given plasmid residing inside the host cell. So these are different types. So we could say based on number of copies, relaxed plasmids normally maintained at multiple copies per cell, stringent plasmids limited copies per cell, fertility F plasmids possess genes 4 pili, hence are capable of conjugation. Then resistant plasmids R contains genes capable of building resistance against antibiotics or poisons. COI plasmids contains genes for coding colicins, proteins that can kill other bacteria. Wait a minute, I would like to explain each of these one by one. Based on nature of copies, plasmids are classified into two types, like relaxed plasmids, which means normally maintained at multiple copies per cell. Stringent plasmids, limited copies per cell. Fertility plasmids possess genes for pili, hence are capable of conjugation. Then later, based on function, three types are there, like fertility plasmids possess genes of pili, hence are capable of conjugation. Then resistant R plasmids contains genes capable of building resistance against antibiotics or poisons. Then COI plasmids contains genes for coding colicins, proteins that can kill other bacteria. Then degradated plasmids capable of digesting unusual substances like toluene, salicylic acid, etc. Then virulence plasmids capable of transforming a bacterium into pathogen. That is virulence plasmids. Then based on ability for sexual conjugation, conjugative plasmids and non-conjugative plasmids contain specific transfer genes that can facilitate the process of conjugation in a bacterium. Then non-conjugative plasmids are unable to initiate the conjugation. They can only be transferred with the help of a conjugative plasmid. So these are different types I mentioned. Then we'll be learning about plasmids in detail. Plasmids. Plasmid PBR322. Plasmid PBR322 was created in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. The P stands for plasmid and R for Bolivar and Rodriguez. Plasmid, Bolivar, Rodriguez. Researchers who constructed it. Okay. Bolivar and Rodriguez. Researchers who constructed it. Then desirable features of PBR322: size 4,362 base pairs, constructed from E. coli plasmid col E1, unique sites ampicillin and tetracycline resistance, replicon from plasmid PBI1, which is responsible for replication of the plasmid, ROP genes encoding ROP protein. ROP proteins are associated with stability of RNAi RNA2 complex and also decreases copy number. The source of ROP gene is PMB1 plasmid. TET gene encoding tetracycline resistance derived from PSC101 plasmid. BLA gene encoding beta lactamase which provide ampicillin resistance. And foreign DNA up to 6 kb can be inserted. Okay, and here we could see the picture of this cloning E. coli cloning vector PBR322 vector showing restriction sites, different restriction sites. PST1, PVU1, Echo R1, CLAL, HIND3, BAM H1, ampicillin resistance, tetracycline resistance, ORI site, origin of replication, ROP site, etc. can be seen. So desirable features of plasmid PUC. Plasmid PUC 18, plasmid PUC 19. Here we could see the picture. PUC plasmids are small, high copy number plasmids of size 2686 base pairs. PUC vectors were developed by Messing and co-workers in the University of California. The P in its name stands for plasmid and UV represents the University of California. Then PUC vectors contain a LAC-Z sequence and multiple cloning sites, MCS within LAC-Z. The MCS is inserted into the LAC-Z sequence which encodes the promoter and the alpha peptide of beta-galactosidase. So insertion of the MCS into the LAC-Z fragment does not affect the ability of a alpha peptide to mediate complementation, while cloning DNA fragments into the MCS does. Therefore, recombinants can be detected by blue-white screening on growth medium containing X-gal in presence of IPTG as an inducer. Then PUC18 and PUC19 vectors are identical apart from the fact that the MCS is arranged in opposite orientation. Then PUC vectors consist of following elements: PMB1 replicon region derived from plasmid PBR322 with single point mutation to increase copy number. BLA gene encoding beta lactamase which provide ampicillin resistance which is derived from PBR322. This site is different from PBR322 by 2-point mutations. E. coli lack operon system. Then ROP gene is removed from its vector which leads to an increase in copy number. Then cosmids. Cosmids are novel cloning vectors which possess the properties of both plasmid and lambda fetus. They were first developed in 1987 by Barbara Hahn and John Collins. Structurally, they possess a co-site characteristic of lambda phages, which is essential for packaging of nucleic acid into protein coat. Scheme of DNA cloning in a plasmid vector and in vitro packaging. So for positive selection, a drug resistance marker, AMPR, a plasmid origin of replication, which regulates cosmid replication and copy number within the Escherichia coli host. Multiple cloning site or polylinker, foreign DNA inserts 45 kb in size is possible. For cloning foreign DNA into cosmid vectors, the following steps are undertaken. Cosmid DNA is first linearized by cutting it with appropriate restriction enzymes. It is treated with the calf intestinal phosphatase enzymes to remove 5' phosphate groups at its ends. This step helps in preventing the recirculation of cosmid DNA. Foreign DNA which is to be cloned is also treated with the same restriction enzyme. Then subsequently, The cosmetic DNA and foreign DNA fragments is incubated with T4 DNA ligase. The desired ligation product is a concatemer of 45 kb foreign DNA fragment and 5 kb cosmetic vector sequence. This concatemer is then packaged into viral particles. Packaging is cause site to cause site, and these are used to infect E. coli, where the cosmetic vector replicates using cold even origin of replication. Then phage packaging. Phage packaging services only to select for recombinant molecules and to transfer this long DNA molecules, 50 kb total, into the bacterial host. 50 kb fragments transform very inefficiently, while phage infection is very efficient. An advantage, the cosmid vector can carry up to 45 kb, whereas plasmid and lambda phage vectors are limited to 25 kb. Increased efficiency to generate a complete genomic library of 10 raised to 6 to 10 raised to 7 clones from one μg of insert. Then genomic libraries of mouse and Drosophila have been generated with the help of cosmid vectors. Then high transformation efficiency. Phagemids. Phagemids contain the filamentanous phages intergenic region comprising the origin of plus and minus strand synthesis and all other cis-acting elements. The minus strand synthesis and all other cis-acting elements needed for the synthesis of ssDNA and packaging it into virions. And phagemids is a DNA-based cloning vector which possesses both bacteriophage and plasmid properties. They carry, in addition to the origin of plasmid replication, a site for origin of replication derived from F1 bacteriophage. Unlike the commonly used plasmids, they differ by having the ability to be packed into the capsid of the bacteriophage. This is because they contain an uri site for double-stranded replication as well as an F1 uri site that enables single-stranded replication and packaging into phage particles. Phagemid vector we could see. For cloning, the phagemid vectors are constructed by following steps. Phagemids are a combination of a plasmid and a lambda phage. Plasmid is inserted into phage lambda genome by means of site-specific recombination termed lifting. They contain functional origins of replication of plasmid and of lambda phage and may be propagated as plasmid or phage in appropriate E. coli host strains. These plasmids can also be released through the reversal of lifting. And phage particles are easy to store and have infinite shelf life. Their screening as plaques by hybridization offers better results than screening of bacterial colonies. Hence, plasmids with cloned genes can be lifted by phages and conveniently handled. Then used in wide variety of biotechnological applications, especially for phage display, it can be used as a cloning vector in combination with filamentanous phage M13. It can be replicated as a plasmid and can be packaged as a single-stranded DNA in viral particles. Then lambda bacteriophage. There are two kinds of lambda phage vectors, insertion vectors and replacement vector. Insertion vectors contain a unique cleavage site, whereas foreign DNA with size of 5 to 11 kb may be inserted. In replacement vectors, the cleavage sites flank a region containing genes not essential for the lytic cycle, and this region may be detected and replaced by the DNA insert in the cloning process. And a larger sized DNA of 8 to 25 kb may be inserted. Then lambda phage. Lambda insertion vectors contain single site for cloning DNA insert. So lambda insertion vectors contain single site for cloning DNA inserts designed to be packaged with or without inserted DNA. Minimum size of packaging 38 kilo base pair, so can only clone in 10 kb or less inserts. Used in cloning cDNA and expression vectors. Then lambda GT10 allows screening using insertion activation of CLG. CI gene in vector has unique echo R1 site. Cloned insert prevents lysogeny, only lytic growth, clear plaques. Without insert, lysogens survive in plaques, turbid plaques. Screen using HFL mutant host. No HFL protease, no cleavage of C2 gene product lambda plus and lambda GT10 without insert always lysogenize. Then if CL insertion inactivated, no lysogeny plaques. Then lambda GT11 has complete lac Z gene cloned with unique EcoR1 site. It is an expression vector where DNA is expressed as a beta galactosidase fusion protein recombinant phage will form. Is an expression vector where DNA is expressed as a beta galactosidase fusion protein recombinant phage will form white colonies and non-recombinant blue colonies with XGAL IPTG. Recombinants can be screened using antibody props. Then potential problems: E insert must be in frame. F antisera must recognize protein produced in E. coli. G proper folding glycosylation may be problem. Then H fusion protein must not be lethal. Kills cells before progeny phage produced. The lambda replacement vectors. A lambda replacement vector has two recognition sites for the restriction endonuclease used for cloning. Then full-length lambda molecule having two identical restriction sites flanked by stuffer fragment. Stuffer fragment is replaced by foreign DNA during restriction cloning. And replacement fragment or stuffer fragment carries additional restriction sites that can be used to cut up into small pieces so that its own reinsertion during a cloning experiment is very unlikely. When cloning large DNA fragments up to 26 kb for genomic DNA libraries, recombinant selection is on the basis of SIE with non-recombinant vectors being too small to be packaged into lambda phage heads. And the vector without the foreign insert cannot be packaged due to the size limitation, smaller than required. Insert size ranges between 10 to 23 kb. PEMBL4. Here is a figure you can see in the stuffer segment of 14 kb that can be replaced by foreign DNA of almost same size. Then PEMBL4. The PEMBL4 replacement vectors have capability to carry up to 8 to 24 kb size insert. The bacteriophage lambda vector have a middle segment accountable for insertion or excision, and this segment can be substituted with the foreign DNA with the aid of two BAMH1 sites found on either side of IE region. External DNA is ligated into the vector and then allowed to infect the bacteria. In the presence of IE region, phage will integrate into the bacterial chromosome and continue lysogeny cycle. But when IE region is disrupted or replaced with the foreign DNA, it will continue lytic cycle and form plaque. Advantages: used for cloning longer DNA fragments up to 20 to 25 kb in size. Possesses greatest transformational efficacy, easier and more convenient for making genomic libraries. An M30 is a filamentous bacteriophage of E. coli and contains a 6.4 to 7.2 kb long single-stranded circular DNA. The virus enters the cell through an F pilus. M13 phage has been variously modified to give rise to a M13MP series of cloning vectors which can be used for cloning of a wide variety of DNA fragments, particularly for the purpose of sequencing through Sanger's method of dideoxy chain termination. Cloning vector of M13MP series have a LAC-Z gene that complements gal host, giving blue colonies. But if a foreign DNA segment is inserted at one of the polylinger sites associated with the LAC-Z gene, it inactivates LAC-Z gene and no complementation is possible. Thus, on transformation, only white or clear plaques are obtained, thus permitting selection of recombinant M13MP plaques. Once the foreign DNA is cloned in M13 vectors, commercially available oligonucleotide polymers are used for copying the insert in presence of dideoxynucleotide so that fragments of different sizes with non-terminae are produced, permitting the determination of the sequence. The reversed order of the restriction sites in polylinker is present in a pair of vectors like M13MP18 and M13MP19, permitting sequencing from both the ends of double-stranded DNA molecule. The extension of the commercial primer or cloned fragment in MIL vector in the presence of radioactively labeled nucleotides will also allow generation of radioactive single-stranded props. So M13MP18 are genetically engineered M13 fragment phages that have the LAC1 gene the lac promoter or operator, and the lac Z alpha fragment gene inserted inside the Z alpha is a polylinker site or MCS. This makes these very useful vectors for such things as cloning and site-directed mutagenesis. The lac Z alpha plus polylinker allows alpha complementation, making it an easily scoreable marker. Then advantages: used as a vector for sequencing mutagenesis, props, lambda, subcloning, phage display libraries. Bacterial artificial chromosome cloning system allows the cloning of a large genomic DNA fragment up to 300 kb in size in cells of E. coli. The BAC vector was first constructed in 1992 and was based on E. coli factor F. The F factor is involved in chromosomal transfer or conjugation. They can exist as extra chromosomal elements. Characteristic features of BAC vectors. The original BAC vector, P. BAC 108L, was based on a mini F plasmid, PMB0131, which carried the following genes: unidirectional self-replicating genes ORIS and REP-E genes, PARA and PRB genes, which maintains the copy number to 1 or 2 per E. coli genome. Multiple cloning sites flanked by universal promoters T7 and SP6, COS-N and LOX-P to permit the linearization of the plasmid for convenient restriction mapping. Chloramphenicol resistance genes for negative selection of non-transformed bacteria. Vector is 6,900 base pair in length and is capable of maintaining insert DNA in excess of 300 kilobases. YAC. Yeast artificial chromosomes are genetically engineered chromosomes derived from the DNA of the yeast. It is a human engineered DNA molecule used to clone DNA sequences in yeast cells. They are the products of a recombinant DNA cloning methodology to isolate and propagate very large segments of DNA in a yeast host. By inserting large fragments of DNA, the inserted sequences can be cloned and physically mapped using a process called chromosome walking. The amount of DNA that can be cloned into a YAC is on average from 200 to 500 kb. However, as much as 1 MB can be cloned into a YAC. Then MAC. Mammalian artificial chromosomes have been studied as a potential avenue for hosting large numbers of transgene in mammalian cells. MAC is about 1 to 5 MB in size. That means a great amount size. And this need the following 3 essential elements for their assembly: a centromere, several replication origins, and 2 telomeric repeats. With the isolation of mammalian telomere and some of the centromeric sequences, it became possible to produce mammalian artificial chromosomes vectors, including human artificial chromosomes HAC vectors. MACs have several advantages over viral-based methods for transgene expression, including a lack of limits on loading capacity, which bypasses tissue associated with insertion into the genome. One area of research in which MACs can be applied is the biomanufacturing of protein-based therapeutics, where reported gene instability in Chinese hamster ovary cells can lead to reduced product titer. MACs can potentially aid in solving this issue by providing alternate hosting sites for transgenes for integration of protein-based therapeutic production. A mammalian artificial chromosome MAC, as the name implies, is a DNA construct that is constructed synthetically and not the same as one of the constituent chromosomes of a mammalian cell. It contains a centromere which allows the cell to split copies properly among daughter cells during mitosis. Because of this property, MACs have been considered as a promising way to host genes within a mammalian cell. 3 types of artificial chromosomes exist: top-down artificial chromosomes, bottom-up or de novo artificial chromosomes, then satellite DNA-based artificial chromosomes. Visual interpretation of the creation of top-down artificial chromosomes. Then HACS were first constructed de novo in 1997 by adding alpha satellite DNA to telomeric and genomic DNA in human HT1080 cells. This resulted in a new mini chromosome contained DNA of interest elements allowing structural and mitotic stability, such as telomeric and centromeric sequences. Instead of 46 chromosomes, the cell could have 47, with the 47th being very small, roughly 6 to 10 megabase pairs in size, able to carry new genes.